So now we move into our afternoon session where we've asked researchers and teams to talk about their findings and results, what needs to happen next, and how we can build on the momentum. So some of these talks are about the impact or long-term benefits, others how we can use this information, and also what happens if we don't. Some of these talks are about how bioheritage projects and programs have worked hard to build trust, engage and develop relationships with research organisations, with iwi, with hapu and whānau, and with agencies, and with individual researchers and knowledge holders, including those co-designed by Māori and communities, industry and end user agencies. So these are pretty much like mini TED Talks. That's what we're going to see on stage now. The presentations, and there are 12 of them, are quick fire, they're lightning, they're about 10 minutes, and a few are only 5 to 6 minutes, and that is my reminder that you'll be sticking to time and the light goes red if you don't, and there is a break for afternoon tea in the middle. But also in the room, excitingly, are a number of invited key personnel from our main stakeholders, the policy makers and shapers, leaders and decision makers. Kia ora koutou, no mai whakapiri mai. So we encourage our delegates to check out the research briefs at the end of the room on the display table there. Anything you'd like to know more about, Keith is standing right there. So are you ready for some quick fire rounds? My favourite part of the chase. And so we welcome our first speaker. On the topic of what is a good decision in biosecurity, please welcome Melanie Newfield from He Tangata, He Taiao, He Ōhanga.
I don't understand what was going through their heads. I don't know all the facts, but for the life of me, I can't understand what they were thinking. Let's face it, we've all thought this. We've probably said it at times. If you've parented teenagers, you might have thought it a lot, and you know that what you've got to do is basically wait till they grow out of it. If you're saying it about a government department, that's more of a worry. And that statement is a direct quote that someone made about a government department. So I'll just move the slides. Kia ora koutou, I'm Melanie Newfield, and I've been working on He Tangata, He Taiao, He Ōhanga, which is a research program looking at risk assessment and decision making, and how to make biosecurity decisions consider a more holistic set of values. And so the piece of work I'm going to speak about is what makes a good biosecurity decision. Now, why have I been researching that? So I was doing some previous work on risk assessment, and it became really obvious that people had really, really different opinions about decisions. So one person could think a decision was absolutely brilliant. Someone else could think the same decision was terrible. And people just didn't have kind of an agreed understanding. And, you know, that quote I gave you at the beginning, somebody else I spoke to used that decision as an example of their good, a good decision they'd been involved with. So here we're trying to improve decision making, but we don't know what a good decision is. Now, before I go any further, and I've just got a pretty picture for you, I want to know what you think before I actually get into my talk. So in your app, if you go to the Q&A, you'll have a poll which is for a word cloud, and I'm asking you to give me one word which you think might be a characteristic of good biosecurity decision making. You put that in, and at the end of the talk we'll have a word cloud which shows what you've come up with. So that way I can kind of just get a bit of an indication from you. So I looked into the decision making research, and I found that biosecurity wasn't the only area having problems knowing whether a decision was good or not. Basically, it's like that everywhere. There's no agreed standard for what's a good decision, and there's an awful lot of research about trying to improve decision making, and no one has any idea whether it's doing any good because they don't know if a decision's good or not. And the only real exception I found was medicine, and they've done a lot of work on what makes a good medical decision. The problem is, it's a good medical decision broadly if it makes one person happy, and that's the patient. If the patient thinks it's a good decision, it was a good decision. It's not a good model for government decision making. We expect a lot more from that. Basically, when your decisions affect a whole lot of people, it gets a whole lot more complicated. So, Basically, what I've been doing, along with my co-authors, excuse the pretty pictures, but I didn't have any words for these slides, is talking about decision-making with people right across the biosecurity system. And I asked people for examples of good decisions and bad decisions, but also their more general thoughts and theoretical ideas on what made a good or a bad decision. So I've spoken with people in central and local government, I've spoken with people in industry, I've spoken with people in environmental, non-governmental groups, I've spoken with people like in air and seaports, and one of my co-authors, Raina Meha, has spoken with people in Māori organisations. And I also dug up some reviews by the Office of the Auditor General and some consultants and various other things, and I kind of added those into the mix. And basically with all that, I've got a series of characteristics of good decision making. And it doesn't kind of work out as an easy recipe, because each of those characteristics has kind of potential for conflict and challenge in it. So what I can do is I've got a research brief at the back which explains them all, but in 10 minutes I can't do that, so I'm just going to mention a couple of them. I'm going to talk about a couple of the ones that are slightly easier to explain and one of the curly ones that really surprised me and I hadn't really thought about. So one of them, it's kind of obvious, good decision making involves the right people and it involves them in the right way. Everyone, of course, has a different opinion on who the right people are, and what works for some people doesn't work for others, and that was really apparent. So the devil is in the details here about who is involved and how people are involved. But what it means is the conversations you're having, particularly at the start of decision processes, are so important to try and get this one right. A couple more, these are kind of at the obvious end. Good decision making is well informed and it's transparent. But even under something obvious like that, there are really different views on what is a good information source. Who do you trust? Who's an expert? And how much information is enough? And people have different ideas about that. And also transparency. What is transparent to one person? And so the person who gave this example of a good decision, then somebody else was like, I don't know what they were thinking. So even in these ones, it's really challenging. And here we get to one of my favourites. Good decision making is well timed. And I'm going to go into this because it didn't show up in the literature when I was looking at that timing was important. But surely it's important in other areas in biosecurity. But what this characteristic says is that decision making needs to take enough time to get things done properly. It needs to take enough time to get the data and the evidence. It needs to take time for the participation. But often biosecurity decisions have some urgency, that there's some situation that's getting worse by the day, or maybe the week. So you've got this tension. What was most interesting to me in this was that when I went through all those review reports, the Office of the Auditor General, the consultants, they were all like, decision making must be timely. And then criticising any slow decision making. When I spoke to the individuals and spoke to this diverse group of people, I got a really much more mixed picture. So some people were talking about timeliness and being prompt, and others were talking about the importance of taking enough time and the drawbacks of rushing. And I'm actually going to read you a few quotes as I do have time. Virtually any time, if you can act early, the costs of an intervention will be less. It was just very frustrating and disappointing to watch because very slow progress was made. One of the main challenges has been the time frame, the real fast time frame to respond. It leaves a lot of our mana whenua behind because it doesn't fit our process. I think you need to be someone who's not flustered, who's not panicked into making a decision just because you're getting a lot of noise or people screaming in your ear at a particular time. I do think you need to make timely decisions, but we often get too hung up on the urgency. And actually some things are not as urgent as people make them out to be, and therefore we rush the decision making, and I think that's a problem. I think you don't want to rush decision making, but you also can't get stuck in paralysis. I don't have any answers and how to get this right, or no magic answers. The crucial thing about decisions is that they're deliberative and that you get into that analytical part of your brain. So part of what we've done is, part of it's about the conversations, and part of it is what we've done is developed some questions to actually get people thinking about how to get this right. So who is setting the timeline for the decision? Why does it need to be made at that time? Are there advantages and disadvantages to deciding more quickly or more slowly? And these decisions could be used by individuals, but also by groups to discuss. They could be used when you are providing input into a decision and providing feedback, and you could use them to help you evaluate past decisions. So what we've done is we have jammed it all on a two-page brief at the back, but we've also put it into a format that makes it a bit easier to read. There's going to be some nice little hard copies for people who actually have desks, but since we acknowledge most of the public service don't, there's going to be an electronic format. We're still working on how to do that. And that's pretty much all I've got time for, except to put the word cloud up. So that is my work in 9 minutes, and so that is fantastic. Is someone going to take a photo of that, or will I be able to get a copy of that later, because that's really cool. And I think there's a lot of what I've seen in there. But yeah, thanks very much.
I love how you drop, and I did it in 9 minutes. Yeah, this is how you do it. There was a couple of words there that got my attention. Unnatural. Fantastic. Okay, cool. This is what the quickfire is all about. Thank you for setting such a glorious standard for us. And now on to our second quickfire session. Please welcome to speak on surveillance, new tools and technologies for surveillance, Steve Pawson from the University of Canterbury.
Aha. So, yep, we're here away from our kids, but school holidays are coming, right? And you can imagine those car trips. And what's a good game to play on a car trip? A ship's coming into harbour carrying a boatload of... What's it carrying? Is it carrying your Easter eggs, your Christmas presents? Is it carrying something you don't want? Who knows? But here we are in New Zealand. And if we make New Zealand the centre of everything, we're pretty isolated, eh? There's a bit of a West Island out there, but if you take a look at it, you know, we're on our own. It's very peaceful. It's very lovely. But if we look into that, we're not as isolated as we think we are. So these are seaport connections or sea connections to us in 2022. And if you start to look more globally with this, and this is a website that tracks about 340,000 vessels around the world. The world is an incredibly connected place. We have been connected for a long time. A lot of these things have been connected for a very long time, but now we're connected faster and in much more volumes. So there's something that we have to accept with this connection, is that trade is never zero risk. Okay, on those ships, in 2021, because I could get the data for that, 850 million shipping containers moved around the world. If we bought 850 million lotto tickets, the stats would tell you you'd win Powerball 22 times over. And in 2021, only 18 people won Powerball. So the odds are worse than Powerball. But what we have to accept is that last year, a million shipping containers came into New Zealand. A 1 in a million chance event will happen. And it's scary to think pre-COVID, more people cross the border than live in New Zealand. So we have to accept that trade and travel is not zero risk. So what are we doing about that? Well, the wonderful folk at Biosecurity New Zealand, MPI, are doing a fantastic job about that. We have a large number of surveillance programs, and they're doing their best, not with increasing revenue for increasing risk with more travel, but in fact with decreasing resources to achieve those goals. And so what we've been doing at SO4 is trying to look at what else is out there that can augment or help improve these surveillance systems so they're more efficient for those threats that we now face. And we've been looking at two technologies, and one of them is looking at DNA sequencing technologies or high-throughput sequencing to overcome the diagnostic impediment. So a lot of those surveillance programs that we run at the moment are focused on individual high-priority species or potentially a few groups of species because diagnostically it's very difficult to have a number of people that can sort through all of those beetles, all those flies, all those Hemiptera. So we chose to look on UV light there. If you look on the figure on the left, you can see the highlighted red box is where the UV spectra is. So across arthropods, there is a fairly general attraction to UV light. It's why we have the zapper trap in the fish and chip shop, right? They all come to UV light. So we have these UV lights on the port. And the data I'm about to show you is from the port of Tauranga, so think concrete jungle. And in that concrete jungle at 15 traps, in this particular set of data taken a year and a half ago, we've got a whole big more data from Tauranga and Canterbury that's literally going through the sequencings this month, so I can't show it to you. But depending on the gene region we chose, we could get up to 1,500 different taxa that we could get hits on. So that's a pretty broad, diverse way of trying to sample what's around the port of Tauranga. But how much of that could we actually put a name on? Okay, so in this concrete jungle, we managed to put names on about a fifth of all of the orders of invertebrates or arthropods that we know in New Zealand. But we could only identify, out of nearly 1,500 taxa, 131 of them to actual species. And that little wheel there, thanks to Wilder Lab, are those species that we could find. Okay, so the power of this resource of using high throughput sequencing with something like UV light trapping is very much dependent on your reference library. Think of it like fingerprints at a crime scene. We can detect all of you that have been in here, but unless you're on the police database, we won't know who you are, right? Same principle here. Okay, so Manpreet's talking next about the power of that reference database and what that allows us to achieve. And tomorrow we'll be hearing from Tiriti-guided who have been using some of these tools about how the reference library could empower them to do biodiversity monitoring. So look forward to that one tomorrow. So the other thing we're looking at is imaging, okay, and taking street imaging. So currently we do high-risk site surveillance where inspectors go around high-risk areas in New Zealand and they do transects looking for potential risks. And that's a diagram there of all of the street trees around Christchurch. So we have a City Council database, 150,000 street trees, a couple of 100,000 park trees, etc. And we're saying, well, could we target those inspectors' effort to go and look at bits of vegetation that we think have changed materially in some way in terms of their health, which could be an indication that a new pest is established. So this is one of our students, Paul. He's up on top of our School of Forestry van. There's a good reason for him being on top of the van. We'll come to that later. But what he was doing is getting imagery so he could create a model. And so his model is around segmentation. So you've probably heard of object detection, where you see those little boxes around an image and say there's a cat and there's a dog and there's a, running around. That's object detection. The cat is in the box. What he's trying to do is segment out of the image the tree. Okay, so his model is the middle image there, and on the right-hand side, as you look at it, is the human annotation of where the cabbage tree is in that image on the left. So his model never saw that human annotation. Okay, this is part of the test data set. So it never saw that, but the one in the middle is what the computer thinks where the cabbage trees are in that image. So we've done that for a bunch of particular different tree species. Now, once you've done that and you know where all those trees are, it's kind of like taking a digital twin. You can start then interrogating and saying, okay, if we segmented out all the elm trees, can we find the ones that look sick with Dutch elm disease? And that's a tractable problem for us in New Zealand because we have that disease here now. But once we know that we can do it, we could start collecting the data for diseases we don't have, things like Xylella that we're all worried about. The one on the right there is that, of course, the thing that comes and kicks you in the butt from behind is the thing that you didn't know was on the boat and you never knew about in the first place because no one else has had it yet. So the question is, how do you find a sick tree? Because trees go through a cycle during the year. They go from fully leaved to deciduous, beautiful autumn colours. There's all sorts of stuff going on. How do you find the sick trees? So the idea is, if you can say, there's an example there of lime trees, Tilia. So we know there's about 5,000 or 6,000 lime trees in Christchurch. And we could create a computer model of this is what the computer thinks a lime tree looks like. And we can keep doing that over time. And then we can go, well, if this particular lime tree moved in its distribution big time, we go, ooh, maybe it's unhealthy. Or maybe a tree tech came and gave it a haircut. We don't know yet. But that's the kind of approach that we're looking at. So, looking to the future, whereabouts with all this kind of thing? So we're starting to have discussions with MPI, we were chatting with them yesterday, around what might a new advanced national biosecurity surveillance system look like? And I think it's going to have to rely on probably two key inputs. One is a DNA-based data source, because we can leverage that power of a reference library. And I think the other might be computer imaging. And the reason why Paul was on top of the van is because we proposed to put them on top of rubbish trucks. How can you afford to put a camera on top of a car and drive it around Auckland every week and image every tree in Auckland? You can't. But if you put it on the rubbish truck, it's going around there every week as well. So a good way of leveraging it. So the more I've been thinking about this, the more I think there isn't another technology out there that's close to adoption that could be used to fulfill this gap and help with these surveillance networks. So we have ways of collecting it. It could be UV light traps for aggregating and flying insects, which is, top left, or it could be that thing next one in. That's a spore trap sucking in spores, so you might be able to get some of the fungi. Maybe we could start looking at wash waters from processing facilities from our crops. Or indeed, what we're already doing as well, building it into the system, is monitoring freshwater, which Lab do very well, and obviously the potential of that is well shown given that they won the Supreme Award at the Biosecurity Awards the other year. Okay, so there's these two technologies, but then they need processing things to go with them. Okay, so in terms of DNA, it's obviously running it through the high throughput sequencing and then through the bioinformatics, and that's a really well proven approach. It's ready to operationalise now. We just need to build that reference library that Manpreet's going to talk about. The pathway for taking the imaging through is probably still a little bit more in the research phase, I think. The machine learning, computer vision, the big use of these GPUs with lots of cooling power, etc., that's still a little way to go. But bringing it in to then having a dashboard of stuff that the surveillance groups at Biosecurity New Zealand and others can use to action and make good decisions in a timely manner. So it wasn't just me that did this, obviously. In fact, I was just the chief cook and bottle washer. But at the top line there, that's all the crew that were involved in the DNA UV light trap stuff. People from Scion, Plant and Food, Manaaki Whenua Landcare, and then on the lower line, people from University of Canterbury and University of Waikato who were working on the image processing. So a big team. Thank you very much.
Thank you so much. Also, the unexpected version of a ship came into the harbour with a cargo load of insects that nobody knows what they really are. And a very good segue and dovetail into our next presentation, thank you so much, because the topic is the Tiriti-guided National DNA Reference Library. And as we just heard as well from Steve, Manpreet Dhami from Manaaki Whenua is our speaker. Nau mai, haere mai.
Kia ora, tēnā koutou katoa, ko Sutlej, ko Biasna Awa, ko Punjab te whenua, ko Jan te roko. No enia ho, ko Manpreet Dhami toko ingoa, e mahi ana i te Manaaki Whenua Landcare Research. Tēnā koutou te whānau by heritage, kua hui mai ne, i tēnei rā, tēnā koutou, tēnā tātou katoa. Kia ora everyone, and first of all, thank you so much to the Bioheritage whānau for this opportunity to present. Our project was one of the later ones, so I have to do the introduction, the progress, and the results all rolled in one presentation. So thank you so much for this opportunity. Just going to figure out the technology. All right, so we've all pretty much seen some version of this image in our careers, lifespans, and if you've looked at the news lately. We know historically biodiversity has been on a steady decline. We're at a point in time where we have an opportunity to really try and address this very big elephant in the room and make decisions and changes around how we eat, behave, and do our mahi to try and reverse some of these declines. And many proposals from better educated people are already out there. But how do we know that this is working? How can we measure success at a large landscape level that can tell us where we are in our trajectory to solve and tackle this very huge problem? One of the very promising approaches is to utilize environmental DNA by which we can stock take our biodiversity en masse. And I won't go into too much detail because I'm sure you're all hearing about this in the news since COVID, how wastewater-based eDNA approaches are helping us understand what's happening around us, and similarly utilizing environmental DNA, just little traces of DNA that we leave behind, that all the biological beings in our environment can leave behind, can tell us about what is out there and potentially how it is changing. So utilizing this really promising tool can tell us a lot about the state of the environment. It can measure whether there are new and emerging invasive species, or the ones that we're trying to control, are they now declining? It can tell us about biodiversity trends of the species we're trying to conserve. It can tell us about novel pathogens, pests, and emerging diseases that we didn't expect but are showing up at our borders, just as Steve had mentioned just earlier. And it can also tell us about tohu species, such as bioindicators for freshwater health. So there are numerous outcomes of this technology that we can apply. Unfortunately, the uptake has been slower in New Zealand than anywhere else in the world. And there are many reasons for that. One in particular being that a lot of our species are endemic. They are only in New Zealand, which means we cannot just leverage off the data that others are collecting overseas. This is something that we have to do ourselves to record and represent our own biodiversity. Also, a number of those species are at risk, which makes it even harder sometimes to track, understand, and develop resources for these species. All this put together is effectively what I refer to as the Aotearoa reference gap, which means many of our taonga, our precious species, are without the required information, be it a DNA barcode, be it physiological data, be it biological data, or even sometimes identity of the species itself. We don't often even know what species these might be. So we have our work cut out for us. Here's just a visual representation of the species that we know are present in New Zealand, but are missing from various reference resources, including a DNA barcode reference. So this one with the very dark black hole of data is a representation of how many species are represented on global databases. For example, for insects alone, around 22,000 invertebrates that we know are present in New Zealand are actually absent in global databases such as BOLD and NCBI. So let that sink in for a minute. When we do have this data, which is shown on the right-hand side, we did this analysis to look at what barcode data is already out there on global databases such as BOLD and NCBI. And what you start to see is that a lot of samples are not identified down to species because the purpose for which these data were generated is not often for biodiversity assessment. So don't need to read all the detail on the plots, but basically it's revealing some of the biases these databases have and that really limit the utilization of these data for these eDNA-based exciting tools. So, as I mentioned earlier, to solve our problem, we're going to have to do it ourselves. And one of the things that really underpins this sort of process is to do it the way we can be proud of in Aotearoa, where we are able to satisfy, to some degree at least, our commitment and obligations to Te Tiriti. So with that in mind, we started a process, and this process has been a little bit kiwiwi kiwawa, I'll just say that hands up already, because we didn't really know exactly what to do, and we asked a lot of people. So really what I will walk you through, which is effectively the progress of this project since its inception in late 2022, is how we took the journey to try and understand what would be the way to develop this reference database. So the project was basically incepted and initiated and funded by Heritage in late 2022, and we ran the very first a Sing Aotearoa workshop, which is a workshop for Indigenous young students to come in and understand genomics, to understand what that means and what opportunities there are for them. And that's where we very first pitched the idea, what would a Tiriti-guided reference database would look like? So asking the young ones. From there, we established a project working group, which was made up of Māori thought leaders, researchers, data managers, curators, biodiversity experts, everybody to come together and start assessing the process of generation of this type of database and what are the key knowledge gaps, where do we don't understand how we could bring in Te Tiriti principles and operationalize them in a way that it's doable as well as respectful. So that was in Feb 2023. Very shortly after, we used these knowledge gaps to launch a Ōhanga series, which was a public forum where we invited overseas and local experts and thought leaders to come and discuss with us and answer really, really prickly questions around data sovereignty and governance and what that would look like in a database that, supported these values. And also we identified some end use cases that would be essential for this reference resource to deliver on, including biodiversity and biosecurity monitoring approaches. We also performed an assessment of international databases that was presented at an additional series of webinars, and we derived governance recommendations from experts in the field of data governance. The Ōhanga series ended in 6 months. It was a huge amount of effort from all our speakers, presenters, experts, organizers who came together to create this huge amount of resource, which is now available online as videos. But also, it attracted a large number of people who attended both live and watched the videos later on. So really generated a huge amount of knowledge and resource that is transferred through the community throughout the motu, and we feel that it's made a bit of a change in that perception around DNA resource, what is possible, what is not possible, because really that first step was to kind of have that buy-in from the community, both researchers and iwi and hapari Māori. From there, the project working group then took all that learning and reassessed that initial process that we'd mapped out and decided what were the technical, governance, and business case priorities moving forward. So very recently, we released a participant survey, which is recently closed. So I'm looking forward to those results to see, get feedback on this process as it's gone on, partially with the technical expertise, but also partially with the social and cultural lens that has been brought over DNA technology in Aotearoa, New Zealand. And this just closed at the end of March. Now, all the hard stuff seems to be over from the outset, but really the smaller core team is now writing it. We're putting the pen to the paper, so to speak, and developing an investment case, which would be part investment, but also part advice to the government on how to build this kind of an enduring long-term resource. Now, a few things that I just wanted to quickly highlight from the Ōhanga series is this. eDNA is really the technology that we need to be utilizing for these large-scale biodiversity and biosecurity monitoring approaches. They are really leading the way, and Aotearoa is being left behind, so we need to really make gains on this and on this soon. Hapari Māori involvement is an essential step from the beginning, and there's just no other way around it. Data sovereignty through provenance attribution is one way we can really enable that Māori data sovereignty within the context of this database. And if I have a moment, I will show later on how to do that, how we can do that. Then this includes legacy data, because data that is already collected is really important. We cannot waste resources to reproduce that data. So the important thing to do here is to reconnect that data back to the iwi, back to the communities, to make sure that they understand what's already being held, and they are responsive to that data as well. again, very loudly within the Ōhanga series, we were told that governance needs to have a directionality to it, where we're heading into Māori control of Māori things, and we have a few ways we can address that within our proposed advice. One is through strong Māori representation on the governance group. This is where the vision of this library and resource will be set, so we need to have that lens over decision making. Any data that is generated by iwi needs to remain with the iwi, and we will be looking at a case study from Ngāti Tura Tinako tomorrow, so that was our first test of this thinking. As well as resourcing for intergenerational development, reconnecting the whānau back to these type of data, back to their ngāhere, and really utilizing this as a way of enabling and empowering kaitiaki of both the resource and the environment. I think I'm really running out of time here, so I'm going to rush through this, but there's going to be live and really good opportunity tomorrow, because I have a few more minutes tomorrow to go over this, to look at how we've been getting introduced to the right tikanga ourselves through the community leaders that we've been working with. And again, this is because DNA is taonga, DNA data is taonga, and so it has to be treated with that respect. Tika and kawa need to be applied where it is appropriate, both in the generation and stewardship of this type of resource. Just a couple of quotes from the Ōhanga series that came up, where I think they've really set the standard high for us, and hopefully we can deliver on it to some degree. We can't do one of these talks without a complicated looking slide. So here is my contribution. This is effectively the blueprint of the library, and I won't go into too much detail, but what I can just point out very quickly is that we have multiple layers of control, and this thing called data dictionaries, where you can, depending on the nature of the data, you can really, at a very fine scale, control who has access, who can see it, who can use it, in what way. And that's really the technological innovation that we are now able to bring into this process, something that has never been considered before. So really we're looking at a globally unique way of generating, storing, and sharing this type of data, which, to be honest, I'm quite intimidated by, but also very happy to say that we've managed to find a solution to do things our way in Aotearoa, but also maintain a link with international resources. So I don't know the pointer if it's working, but basically the little wee thing in the orange color is all I want to talk about today in this very complicated diagram, which is the bin structure. A bin structure is a secondary data derived from all the primary data resources. So things like DNA sequences, geolocality, things that are of importance to us never leave Aotearoa. They stay in country, they stay in place, but we can derive sort of an imprint of what these data mean, and that can be shared globally, because that is data that we can help train others, but also train our own data to make these kind of global biodiversity assessments that we so badly need today with the scale of the issues we're dealing with. Again, there's a few more notes, and I'm more than happy to discuss with anybody who's interested in these, how the data comes in, how the data leaves, what end users are there, what data does never leave. So there's a whole range of complexity within this diagram that I'm not going to be able to tackle in this very short presentation. Finally, just a few notes to wrap this up. I truly believe I've seen a lot of databases, I've interfaced with a lot of databases. My whole career has been about doing DNA-based research, so I can put my hand on heart and say that this is really, truly a globally unique endeavor. We just haven't really considered these approaches previously because we didn't have to. But within Aotearoa, we have to, and I think that is the way forward for us, and to really show the whole world that how can we respect our cultural values as well as do good science. So here we are leveraging technological advances to deliver on our Te Tiriti commitments. The decision dictionaries that I spoke very briefly about, but happy to talk in detail, are enabling this type of fine control where iwi, hapu, who are participating, can have that power to control, to change, and if they want to share their data. They have all of those options in their hands. And that kind of data sovereignty at individual discrete datum level is just not available anywhere else. This repository, and I think Steve has already mentioned it before, is essential for us to apply eDNA technology at species level quality, at a resolution that is required for making decisions. eDNA is already being applied at levels where we can understand trends, but if we want to enact change, if we want to enact restoration outcomes, we need to understand these data to a higher level, and that's what this resource can underpin. Biodiversity monitoring, biosurveillance, and biosecurity are two key outcomes, but there are many, many uses. This library is also going to tell us what new species are out there that we haven't yet even discovered. So really, this is a resource that underpins a lot of new knowledge that can be derived, as well as addressing some pretty major concerns. Finally, we can't do it without the people. It's going to take buy-in from Hapari Māori, from thought leaders, from data managers, from scientists of a variety of ilk. So really, we need everybody, all hands on deck for this. If anybody is interested in knowing how much it is going to cost, they can come and find me in the break, and I can whisper a number into your ear. But really, beyond that, we also need really strong leadership and governance of this resource. This is a resource for all of Aotearoa. It needs to be enduring. It needs to have those te tiriti principles really embedded into that governance for this future resource. And I think with that, I will just say thank you to a very large group of people who have been absolutely instrumental. Some of you are in the room in the development of this vision, in the development of this thinking, and I'm just a spokesperson. Holden opened the meeting this morning, and we were all lucky to see him, but I've been trying to find him since then. He's the co-lead of this project with me, and absolutely my moral compass when we have to make some tricky decisions, so tautoko to him all the time. But yeah, the Ōhanga series videos are available online if anybody's interested in any of the topics that we addressed. Some of the speakers are also here who contributed their expertise. And just a very last note of thanks to partners who've been really testing this thinking with us on the ground and giving us live feedback around what is operational and what is not, and what is of value and what is not. And that has been absolutely crucial at refining our thinking, and that's Ngāti Tura Ngāti Tanaka, who will be speaking tomorrow. So thank you, everyone. Ngā mihi nui. That's me.
Tēnā rau atu koe. Thank you so much, Manpreet. And a complicated slide is what we all need. And as you say, you've got more time tomorrow, about mid-morning as well. Do like the quick reference to kiwiwi, kiwawa. Do we all know that term? Kiwiwi? Kōrero mai, kiwiwi? Kiwawa. And that means all over the place. Anyone been there? Scattered places, kiwiwi, kiwawa, going all over the place, but not us. We're back into the quick fire. Quick fire session number 4. Please welcome to speak to the topic Beyond the Traps, Community Values Drive Choices of Predator Management Tools, Ali Palmer and Izzy Busby from the University of Canterbury. Kia ora. Kōrua. Big hand.
Kia ora, everyone. I'm actually from the University of Auckland. Sorry.
Is this the tomorrow's presentation?
Oh, it is.
Can we have the other presentation?
Oh, you're good noticing. Yeah, we're talking twice, and this is the long version. Do we have a short version available?
Is it within tomorrow's one?
Sorry? What do you mean?
This is the long one.
This is the one for tomorrow, yeah. Yes, I mean, we could talk for a long time now, but I get the feeling everyone will want afternoon tea, so.
Okay, just no.
No?
You're gonna feel free to skip through and go, this is the preview, we're having a premonition of tomorrow.
All right.
Sorry.
No worries. And how do I click through if I need to? Do I just... Oh, this thing. Okay. Big green. Big green, thanks. All right. Well, kia ora everyone. My name is Ali. I'm actually from the University of Auckland. I'm a social scientist, although I'm weirdly in a biological sciences department. And I should say that I'm funded by Predator Free 2050 Limited. And Izzy, who's actually been leading this project, I'm sorry to be the one introducing us, is from the University of Canterbury, the Ngaitahu Centre. And we have been doing some work for the SO5 strand of bioheritage looking at social dimensions of invasive species management. Excuse me, something's happening with my voice. So essentially studies from around the world have suggested that, oh, brilliant, thank you, have shown that social acceptability is crucial to invasive species management, and a lack of social acceptance can completely undermine projects, let alone the question of should you actually proceed with a project if your community is not behind it. And two really important sources of social conflict in many examples from around the world have been the method of lethal control. So think about 1080 in New Zealand, for example, and also mistrust of government institutions that are often leading invasive species management. But for Predator Free 2050, something quite unique is happening where community groups are actually often leading on delivering projects rather than government agencies. And so in theory, these groups are kind of closer to their communities and might be a lot more trusted. And so they might then face less opposition than government in general, and also in particular to lethal control methods. Possibly if a community group is much more trusted, then actually that controversy around method disappears. That's the theory anyway. But we don't really understand how community groups build and maintain trust, and whether they do actually find that they're able to kind of diffuse any potential controversies around lethal control methods due to that greater trust. And so we wanted to find out more about these questions. And to do so, we conducted a series of focus groups, or actually Izzy conducted the focus groups, and the two of us designed a survey to send out to community groups to ask them about how they select their tools, particularly around thinking around social acceptability of lethal control methods, and also just more generally how they seek to maintain social acceptability. And today we're going to share just a couple of key findings. Tomorrow we're going to talk for slightly longer in more detail. Did that work? Big green button? Thank you. Very briefly, as is probably obvious to everyone, some methods of controlling invasive species are way more accepted than others. When we asked community groups about this, totally unsurprising, kill traps were by far the most accepted, toxins were much less so, and interestingly, leg hole traps were down the bottom there. Lots of different factors came up as important for how these tools are chosen, with efficacy ranked as the most important consideration when thinking about what's important to look for in a lethal control method. But focus groups especially showed that method choice is really, it's just fundamentally constrained by social acceptance. So that low ranking of toxins, for example, was often groups saying, it's not that I personally object to toxins, it's that my community won't accept toxins. So very often there was a discrepancy between what people would personally want and what they felt would be socially acceptable. And in general, actually, we found a lot of groups indicated they would personally like to use toxins, but they felt that they couldn't, essentially due to social acceptance reasons. So this quote here from Brian, not his real name, we use pseudonyms, it's pretty representative of what quite a lot of our participants said. And this demonstrates really that despite good relations with communities, these groups often still struggle with acceptability of tools. In particular, we suggest that essentially historical cases of conflict with government agencies can stick to the tools. So essentially, even if the implementer, the community group, is much more trusted, that doesn't mean that they can then use a method that's been contentious in the past, even if they kind of didn't create the contention. And it really highlights how important it is to just not muck up the social relationships in the first place. Had the bad blood around toxins, for example, never happened, perhaps these more highly trusted community groups would actually be able to use them now. So given this situation, groups essentially took two quite different approaches. Some groups did attempt to kind of do some educational persuasion with their communities around, hey, we'd really like to use this method, can we, talk to you about why we think it's okay? But others essentially sought to understand the tools that their community would accept and essentially said, no, we won't ever attempt to persuade or change that. Essentially because the sense that trying to change people's minds would itself be so contentious that it was better not to go there. And so for some, even talking about toxins could undermine social acceptability. So see the quote from Jane here. I'm now going to hand over to Izzy to talk a little bit more about some of our other results.
Thank you, Ellie. Kia ora koutou. So, how are the groups actually maintaining their social acceptance? So stakeholder engagement has been recognised as essential for some time. However, it's often thought of, it's an afterthought, especially in invasive species management, because of that pressure to respond really quickly, right? But here, groups emphasise the importance of engaging or co-designing with stakeholders in mana whenua before the project even begins. So for example, we have this quote from Nico, where he explains, if you get people up and running too early without creating the correct relationships, they run with it. And once you've gone too far, you can't backpedal. Another way was the regular and transparent communication that these groups had. So groups described their communication as a way of maintaining that social acceptance with their community. So as we have this quote from Wiremu, which he explains, our comms, our pānui, our newsletter, and Facebook page have created awareness in the community of what is actually going on, because not many people understand and saw it as just killing animals. And then lastly, we have this positive framing. So groups expressed and ensured that they were framing their operations with a positive and outcomes-based approach. So for example, we have this quote from Tane who says, it's always important that your objective isn't to kill all possums. Your objective is to increase the vegetation, health, and have it sort of framed in that perspective. So among other things, the groups that we consulted often wanted more information on cost effectiveness in their local environment, and so for that reason, the other part of our team in SO5 is developing an online tool aimed at helping groups understand that in an accessible way. So that's our TrapSim Plus I have up here. So the tool allows the users to choose a number of parameters, such as their area on a map, along with the traps they want to use and the pests that they're targeting, to produce a comparison of the cost effectiveness of different methods. But as our research demonstrates, these decisions must be interpreted through the lens of social acceptability. So while the tool indicates which methods will be cost effective, they may not be seen as socially feasible. And so we leave that up to the individual groups to decide, given that their social contexts are unique. And then moving forward, so how does this help us move forward with Predator Free 2050? Well, the technicalities of the movement are important. So what traps, how many, where we're placing them. But it mustn't be forgotten that this is also a social movement, and predator control cannot happen in isolation without communities on board. And we've heard that time and time again this morning. It's ground up. So our results highlight that community groups recognise this. They operate accordingly, particularly because of that historical controversy that Ali talked about. And they engage with their stakeholders. They engage early, they regularly update the wider community, and then they communicate their control in that positive light. So that's all from us, and we'll talk a bit more in depth tomorrow, but we'd like to thank the rest of the team, particularly Chris and Nikki at Manaaki Whenua, and Andrew Gormley, also at Landcare Research, for his kind of creation and development of this online TrapSim tool, and of course BioHeritage for the funding. Thank you.
Kia ora kōrua, awesome recovery. Sorry about that you had to go with some technical difficulties. And of course, that Ali Palmer is actually from the University of Auckland, Tamaki Taumatarau. Now we are up to quickfire number 5. Please welcome to speak to controlling invasive invertebrates, Phil Lester and Ocean Mercier from Victoria University. Kia ora kōrua.
Tēnā koe Stacey, o tira koutou katoa, e mihi ana māua, kia koutou katoa te whānau whānui e tiaki nei, e tō tātou koe ora tuku iho ngā mihi, ko Ocean Mercier ahau no Ngāti Porou, ko Phil Lester tēnā he no Ngāti Pākehā. And we're going to do a bit of a tag team. I'll pass over to Phil soon, and then I'll step in to continue on this presentation, in which we are interested in socially acceptable, following on from our last speakers, cost-effective and highly targeted next-generation technologies, tools and strategies for invertebrate pests control. We are part of the SO5 invertebrates team, and I'll now pass on to Phil.
Thanks, Ocean. Kia ora. As Ocean said, it's our privilege to be here and talking today, and it's our privilege to represent really a large group of people that have been involved in this part of the challenge for the last 10 years or so. That was our goal that we started in Tranche 1 10 years ago. It's hard to believe that it's that long ago and time flies. And it is, as quoted there, as Ocean stated, to develop a new way of controlling invertebrate pests that is socially acceptable, cost-effective and highly targeted next-generation approaches. Initially, when we started this, you see us pushing the button, there we go. We wanted to work on some pests. So what pests? We're going to have to not work on every pest in the world that we decided. So we decided on a couple of pests, primarily wasps in the first tranche, to work on those. Those are a massive problem in our honeydew beech forests here in New Zealand, as many of you will be aware. One of the world's worst invasive species to invade New Zealand. And the second pest that we've been working on as well is varroa mites. So those varroa mites are a key pest of honeybee populations in New Zealand, one of the major forms or causes of overwintering mortality for honeybees. Initially, what we did was looked at, had a large number of groups involved. This is in Tranche 1, so 2014 to 2019. So there were a large number of groups led by these organisations and with collaborators out as well. So there was plant and food research looking at novel use of artificial pheromones to control populations. There was Manaaki Whenua looking at using mites to deliver pesticides or pathogens in a novel way, or a novel approach. There was a program led through us with lots of collaborators looking through gene silencing or synthetic RNA for targeted pest control. Ōhanga University had a role in terms of genetic modification of pest populations that was either manipulated by natural genetic deficits in populations out there, or in breeding genetic modifications into populations. And University of Auckland were looking at some novel ecological interactions between pest species and invasive species. We went on to look in Tranche 2 and just 2 of those technologies. So we thought at the end of the Tranche 1, well, what has got the most potential to get us to our end position, to get us to our end goal in terms of getting a novel, new, targeted pest control technology. And these are the 2 that the groups decided on: gene silencing for RNA for targeted pest control, or genetic manipulation, modification of pest populations. And I'll talk more about that one tomorrow. Today we're going to focus really on the gene silencing approach. So what is gene silencing? Gene silencing or RNA interference is a stop button, really. It's not genetic modification, and there's no genetic modification about it. It's an essential, stopping the production of an essential protein for an animal species using double-stranded RNA. What's going on here is, in this video, is a delivery of those products. So on your left there is the standard approach of utilising pesticides for varroa control in this case. Those are the strips that he's pointing to there. And on the right is our treatment for varroa mites, looking at using double-stranded RNA in little pouches that we give to honeybees, and the honeybees feed on that and take it and then distribute it throughout the hive. What you can see there is actually really quick to deliver by the beekeepers, so it takes about actually half as long. The bees quite like it, so they'll land on the pouch as is going on there. They will then drink from the little perforations in that pouch, which is what those two bees are doing. It's in sugar water, and then distribute it throughout the hive. So it's a quick product to deliver to the hives. The bees seem to like it. The bees don't seem to be affected really by it in any ways. It's a food source really for what's going on in those populations. And it's quick to deliver. So as about half as long there as the regular standard pesticide approach. This is lots of field trials throughout the program. So we had field trials that were going on in the Manawatu, up near Fielding, and down in Canterbury as well, using some hundreds of hives. Here are some beekeepers that are involved in this trial, and they're putting those little pouches within the hives. So that's what they're doing there, and then the bees feed on them, and away they go. What did we find? So this is trying to summarise 5 years of work very quickly, but our work has shown that this can be a highly targeted and quite species-specific way of controlling the key pest, in this case, varroa mites, in beehives. So it can work, and that's very much the case in the laboratory populations, where we can knock down mite reproduction almost entirely, and depending on the dose and a number of different factors, we can do similar things in honeybee hives, which you'll hear about a little bit more tomorrow. Seems safe for honeybees, so honeybees don't seem to be affected. We've had a large program as well in terms of looking at, well, what does it do on other species within the hive? So the moth there is a wax moth. That's something you might find in honeybee hives. It's another pest, and it doesn't seem to affect those. Where we've got to in this program is now to be able to get to a point where, okay, we've got potential, at least, to start the registration process for this product as a new pest control approach, a new mode of action, really, for controlling pests using the varroa mite as an exemplar species in New Zealand. We're working with the company in discussions and with a number of different groups around starting that even potentially this year with the US collaborator that's involved in making this product. It's a new. I think it's a pretty exciting outcome. It's a new mode of action that could be, and has potential to be used for a wide range of other pests as well. And I'll pass it over to Ocean.
Tēnā anō. So varroa mite destroying honeybees and their kāinga are environmental, social and economic concerns for Māori and non-Māori alike. And we spoke with 62 tangata whenua with different backgrounds and interests to gauge their views on not just this particular technology, but traditional and contemporary pest control methods, including gene-based technologies, and we'll expand on other gene-based technologies tomorrow. 20 of the ones that we spoke to were Māori beekeepers, who we were asking specifically about their views on this technique for controlling varroa mite. And we're focused on tangata whenua views for obvious reasons, I think, to ensure representation and voice of tangata whenua, because we're, Tiriti partner. We've got a lot of, hundreds of years, as we heard from Tami this morning, but also partly because Māori articulate concerns based on tikanga and te ao Māori, in addition to all the standard mainstream concerns regarding safety, efficacy, unintended consequences, etc., etc. So we must first acknowledge in this conversation that pest control is not new to Māori, nor is innovation in pest management. Elimination strategies were key to successful kumara cropping and agriculture, and our pest management strategies were numerous, with some customary methods aligning with contemporary strategies as well as being novel in their own right. Again, as we've heard in other presentations today, turning pests into products, for instance. And secondly, kaitiaki are responsible for protecting taonga species, so invasive pest management is a continued priority activity for tangata whenua. Māori have long expressed their concerns publicly about destruction of bird habitat, for instance, as documented in several 19th century accounts. Given this and those hundreds of years of practice here in Aotearoa in pest management, tangata whenua should be at the forefront of contemporary discussions, and Māori-led approaches to pest management add much in their holistic, innovative and also specific approaches. Which leads me to my final point, that concerns and aspirations for new tech expressed by people that we spoke to are highly specific across a range of interest areas. This particular technology ticked a lot of boxes for tangata whenua who are managing bee colonies. They liked its targetedness, the specificity, that it amplified upon a natural process or a process perceived to be natural, presented as humane, and assurances that it didn't break whakapapa lines. Criticisms arose of the name, RNAi, pretty a difficult one to work with there, but gene silencing was a little more palatable. And also the need for seasonal application, for one non-beekeeper noting that in comparison to things like gene drive, gene silencing was too stop-start because of the seasonal application needs. Just to give you a snapshot of some of the thoughts that came from our interviews, aspirations-wise, Māori beekeepers also had unique challenges and constraints and opportunities, including uplifting whānau and hapu through employment in whānau businesses around honey production, wanting to use their uniquenesses to gain an authentic edge over competitors, and on the other side, seeing encroachment upon their cultural and intellectual property by other businesses. So the opportunity for a new targeted control to help with varroa mite was welcomed, but it's one of a number of concerns that Māori beekeepers were dealing with. I think I'll just move on to finish there by acknowledging the fantastic team that have worked on this project, 4 of whom gained their master's degrees on this program, ka pai koutou, especially acknowledging Simon and Alan, who recruited, surveyed and interviewed Māori beekeepers specifically, supported by Tere. We're grateful to others who've helped along the way, especially those in bioheritage, Stacey and Waimirirangi, whose mahi has supported our communication work with our participants, who we also very gratefully acknowledge. Tēnā koutou.
Kia ora korua, Phil and Ocean, and now just 2 more quick fire speakers, and then our job is to eat chocolate just for a short amount of time. So kia kaha tātou. Our next speaker is speaking to the topic of dialogue on complex socio-environmental issues, and it is Marie McEntee from the University of Auckland. Nau mai, hoki mai.
Kia ora koutou, ko Marie McEntee tōku ingoa, ko Waipapa Tamata Row, the University of Auckland, aho, ngā mihi mata kiwikui. Today I present a methodology that we have developed for engaging people in dialogue around complex and contested issues. We developed this methodology for our bioheritage funded project, which we began just over a year ago, to engage New Zealanders in conversations about the use of gene technologies for environmental purposes. However, this methodology, which I describe in this presentation, could be used for any contested issue. Indeed, it could be used for the one on pest technologies for community groups. Tomorrow, my project co-lead, Dr. Fabian Mitvicki, presents some initial insights from the project engagement with the wider public and also with Māori communities. I would like to take this opportunity to acknowledge our partners, Te Tira Whakamātaki, who have been leading the project's conversations with Māori communities. In this presentation, I am mainly going to concentrate on our public engagement as we do not have prior relationships with these communities, so they present a particular challenging cohort to engage. I think we all agree that we live in a complex and often contested world, whether it be in regards to the closure of forests for biosecurity reasons, or in the application of novel innovations such as genetic technologies. Our world is complex. Jerome Ravitz, who is a philosopher of science from Oxford University, states that in complex issues like genetic technologies, facts are uncertain, values in dispute, stakes high and decisions urgent. And the framing of the problem involves politics and values as much as it involves science. While we typically acknowledge the technical uncertainty of gene technology, we recognise what makes this more complex are the social uncertainties that stem from the diverse and divergent perspectives that emerge from the use of gene tech. The science and society literature is emphatic. If we are to tame this complexity, we must engage society in deliberative discussions, and this engagement needs to be open, inclusive, responsible, and collaborative. With gene technologies, we cannot simply seek a social licence to operate to gain society's approval for technologies as though it was almost a tech transfer approach. Instead, we must embark on responsible innovation, where there is continuous engagement of the public along the entire innovation pathway, and careful assessment of the possible risks, including impacts on values, morals, social relations, health, the environment, the economy. And specific to the context of Aotearoa, where decisions need to be responsive to Te Tiriti, impacts on Māori culture, values, beliefs, and practices. So any engagement around novel technologies must firstly seek to prevent the divide that often exists in this space. But the question remains, how do we do this? How do we effectively engage communities around complex and contested issues, and particularly in conversations with the wider public that often come to these conversations with very strong positions? Our project has sought to find a way to answer this by developing a methodology for productive dialogue, which we have utilised for our engagement process. We are using this methodology, which I'm going to describe in a minute, to ask, how do New Zealanders think and feel about the potential use of genetic technologies for the environment? How might genetic technologies relate to people's visions for the future of New Zealand's environment? For what issues, problems might genetic technologies be acceptable? With what guidelines, rules and limits? And who would people trust to oversee decisions? Tomorrow we're going to look at some of those, but today I'm concentrating primarily on the methodology. So this is how we've gone about doing this. To meaningfully engage people, we have used a three-stepped approach, a very purposeful approach. First, to explore, to both listen to and gather perspectives, then to refine, to develop scenarios, and finally to deliberate, to build consensus, to co-develop a path forward. The explore phase focuses on listening and gathering people's perspectives to inform the development of the next two phases. So we start by listening to the community. However, in the public conversations, as we have been working with communities we have never met before, we need to build relationships. And we need to also overcome the anxiety that people have about engaging in conversations about gene tech. To achieve this, we developed a series of purposeful games to build people's understanding and confidence to engage in the strange and alienating language and world of gene tech. We are making these games available at lunch tomorrow, so you can see how purposeful games can be used in some contexts to overcome people's anxieties around engaging in complex issues. We welcome you to join us at our session tomorrow. You can enjoy us too if you like. Once we have listened to communities' perspectives, we embarked on phase 2, which was refine. For this phase, we have engaged with a number of specialists, many of you who are sitting in this audience today, and have also engaged with interest groups, including GE-free groups, to inform the development of scenarios for our deliberative phase. We have found that scenarios need to be real as possible, and they need to cover the complexities of each problem, and they also need to deal with the current and future management tools. During this phase, we have developed 4 scenarios for our public deliberations. These are, and you just heard the first one, varroa mite and myrtle rust, both RNAi technologies, so gene silencing, so not technically regarded as genetic engineering. And we are also dealing with wilding pines and rats, both genetic engineering technologies, one very real, and one really in its infancy, as we're somewhat further away from mammalian predator control. TTW has used 5 different gene technology scenarios that have specific impact on a range of cultural values, and have discussed these in focus groups, drawing on expertise of kaumātua to inform their discussions. In our third phase, deliberation, we actually seek to build some consensus. For the public conversations, what we are doing is we are positioning our scenarios in a complex landscape. You will see the map here on the slide that we have developed for this phase. This positions the scenarios in the political, social, cultural, economic and regulatory landscape that these technologies operate in. We cannot divorce these conversations about gene technology from this complexity. We also give our participants a range of the perspectives that emerge during phase 1. So the landscape has the voice of people in it. This relaxes people because they can see themselves in some of these voices, and it exposes them also to new voices that they hadn't even thought about. These voices inform the deliberations that follow the small group discussions. People are asked to reach consensus, if at all possible, to write down what drove their decision and what guidelines or rules they want to guide the implementation of any new technologies. The strength, therefore, of this 3-phased dialogic process lies in the fact that, first of all, it starts with listening to people's perspectives to inform the other phases. Secondly, it seeks to overcome epistemic anxiety that people have about even believing they can engage in gene technologies. And this is particularly important for public discussions. Next, it enables strangers to be able to work together. It also enables voices that are not usually heard to also be heard. And it values the many, many multiple perspectives that exist around this. It also seeks to create a safe place to engage. And very importantly, rather than simply asking people, do they or do they not agree, it gathers nuanced understandings about their perceptions of gene technologies. This process of explore, refine and deliberate provides a robust methodology for dialogue in any contested issue to gather perspectives, identify obstacles, possibilities and pathways to inform good policy making. When stakes are high and decisions are urgent, and the framing of the problem involves politics as much as it is, and values as much as it does science, we need what Sheila Jasanoff from Harvard has said are the technologies of humility, founded on the process of deliberation and dialogue to engage people as active participants. If you wish to discuss further, please come along to our session tomorrow. You can also look at our website, and Te Tera Whakamātaki is here also to talk about the Māori deliberation that has been undertaken. Thank you.
Kia ora, Marie, thank you so much for the invite for tomorrow as well. Can't wait to enjoy you and all that we offer tomorrow. But it was fantastic today as well. Tēnā rawa te koe, Marie from Waipapa Taumatarau. I liked hearing your summary there, technologies of humility, and it reminds me of a whakatauki ko te whakaiti te whare o whakaaro nui. Very similar idea wrapped up in a whakatauki from Dr. Whare Huia Milroy. So we are so nearly there. We are nearly at our afternoon tea time, but please welcome and give great anticipation and appreciation for our final speaker for this side of the afternoon tea. So speaking to anticipating Aotearoa's weedy future, please welcome Angela Brandt from Manaaki Whenua.
Tēnā koutou katoa, ko Angela tōku ingoa. I'm an ecologist at Manaaki Whenua and help coordinate the weed invasion and ecology research area. And as you can probably tell from my title, I'm talking today a bit more about what are our next steps. So the global accelerating impacts of invasive species were demonstrated just last October with the release of this major international synthesis, which many of you are likely familiar. And why does this matter to us in this room? Well, invasive species are one of the 5 major drivers of biodiversity loss worldwide, but they also have many impacts for people. And a bit more locally, in 2021, the Parliamentary Commissioner for the Environment released a report that recommended some needed improvements to Aotearoa New Zealand's biosecurity system to enable us to adequately protect our native ecosystems from the threats due to weeds. And this report involved heaps of input from local communities and those who are working on the ground to do the important mahi of protecting those ecosystems through weed management. Chapter 3 in that report highlighted how the weed problems that we face are going to grow in the future, which means we need to be acting now to prevent or reduce these threats. So there are already tens of thousands of non-native plant species present in Aotearoa. Approximately 1,800 of those are naturalized, which means they have self-sustaining populations in the wild. And approximately 400 are currently managed as weeds. But it's estimated that 20 additional non-native plant species are naturalizing every year, and very few, if any, of the current weeds that we're managing are actually established at the full extent of where they could grow in Aotearoa. Moreover, climate change, land use change, and other environmental and societal changes are influencing weed invasions and can influence their impacts. These changes are likely in the future to be exacerbating the weed problems that we currently face with the weeds we're currently managing. So these 2 factors are informing 2 research pathways that we're pursuing. And by we, I mean those of us within Manaaki Whenua, but also our collaborators across universities, other research institutions, and stakeholder partners. So first, we need to support a shift to proactive weed management in Aotearoa. We need to be tackling weeds earlier on this invasion curve. We need to bring together ecological, socioeconomic expertise, mataranga, to provide the tools, information, prioritization, and coordination required to enable those who are managing weeds on the ground to act before a species is becoming widespread. We have research teams and stakeholder partners who are keen, have the ideas, have the capability to do this, and we're currently seeking to secure funding so that we can make it happen. And many of those team members and stakeholder partners are actually in the room, so it's excellent to see you all here and the excellent mahi that you're doing across wider bioheritage space. Second, we need to identify what management challenges could arise in the future, especially given the environmental and societal changes that we're seeing trending now. If we do this, we can actually undertake research now that is going to be more likely to address the questions and challenges that are coming our way in 10, 20, or 30 years' time. This was the focus of our challenge project, where we were involved in building some scenarios of what Aotearoa might look like in 2050, evaluating how those scenarios could impact weed invasion, and identify the types of research that we could do to actually best address the weed management challenges that could arise through those. And Jennifer Bufford's going to talk more about this project tomorrow, so there's a little shout out, and please do come listen to that talk. So why does this matter? Well, weed invasion can be insidious. We often don't see changes or impacts in the first year or 2 years or even longer after invasion. But if we wait too long, weeds will grow out of control. We know we can achieve much more, and it can be lower cost on our wallets and our valued bioheritage if we act sooner rather than later. So I encourage you to talk to us about how the research we've done and what we plan to do next could help you and all of Aotearoa actually be ready to tackle this potentially very weedy future head on. Thank you.
Kia ora, Angela. That was just like doing a skid into the end of the car park. It's going, and she's finished. Ta-da! Fantastic. Thank you so much, Angela. And all of our quick fire speakers, please put your hands together. Because now we have some very exciting information about afternoon tea, which will bring us back to then another 5 quick fire speakers and our fantastic keynote speaker to end our day as well, Matua Kevin Prime. But I must tell you about this cuppa and chocolate. So afternoon tea refreshments at Crazy and Ambitious Tour number 4 are sponsored by Bioprotection Aotearoa, which will probably make you just burst into applause. As you know, a distinguished National Centre of Research Excellence hosted at Lincoln University, uniting 7 universities and 4 Crown Research Institutes, and a shared mission to train future bioprotection leaders and address environmental challenges in Aotearoa, New Zealand and the Pacific. Thriving on multidisciplinary research, Bioprotection Aotearoa develops new and innovative solutions to safeguard our productive and natural landscapes from pathogens, pests and weeds, so it doesn't get a weedy future ahead of us amidst a changing climate. So we would really like to thank Bioprotection Aotearoa for their commitment to environmental stewardship and innovation, and for their support for this conference. And you want to enjoy today and also tomorrow, because they have sponsored afternoon tea, where Bioprotection Aotearoa are providing a very sweet treat. So as well as tea and coffee and other drinks at today's afternoon tea, we also have chocolates provided by Beneto Natural Foods, which is a small company in the big world of chocolate. There's chocolate, then there's chocolate, started in a test kitchen and garage in suburban Ōtautahi, Christchurch. You will know Beneto from those beautiful bird illustrations that are on their packets as well. And they are also organic, fair trade, craft chocolate. So please try the new flavours. It really is your job as you enjoy this afternoon's refreshment break. Just a quick kara kia kai. I'll repeat as Kai Whare said. E rongo ho mai ngā tipu hei whakaki te tinana hei o ranga auweke hui e tae ki e. Enjoy, and we'll be back really soon about 3:10. Kia ora.